Hi, my name is Taylor McLaughlin, and I plan to pursue a career in biochemistry with a focus in medical research. Today I will be performing a DNA extraction using a strawberry as a model organism. In this lab demonstration, I'll be breaking open the strawberry cell and separating their components and isolating their DNA so it becomes visible to the naked eye. Although this experiment is simplified and uses household materials, it reflects the same core biochemical principles scientists use in professional laboratories when studying genetic disease and developing new treatments. As I mash the strawberries, I'm physically breaking down the cell walls. DNA is stored inside of the nucleus of the cell, so the first step in any genetic research process is breaking open those cells to access the genetic material inside. In this, I start by adding a half a cup of water, and then I add a teaspoon of just normal salt, and then I add about a teaspoon and a half of dish soap. This solution helps release the DNA from the strawberry cells. The dish soap breaks down the lipid bilayer of the cell membrane and nuclear membrane. Since those membranes are made out of fats, soap is able to dissolve them. The salt helps neutralize the negative charges on the DNA molecule, allowing them to clump together later so we can actually see them. After the strawberry mixture and solution is fully combined, I then move on to strain the mixture. This step helps remove larger pieces of cell debris, leaving behind a liquid that contains dissolvent DNA and other cellular components. In professional labs, scientists use centrifuges and filtration systems, but this is a simplified version of the demonstration, but it's the same concept. I am now adding very cold rubbing alcohol that was left in the freezer for 30 minutes. I do this because DNA is not soluble in alcohol, especially when it is cold. As the alcohol forms a layer on top of the strawberry solution, the DNA begins to precipitate out. The white, cloudy strands forming at the top are DNA. This is the genetic blueprint of the strawberry. In medical research, scientists extract DNA to identify genetic mutations, study inherited disorders, and develop treatments tailored to a patient's genetic profile. Understanding DNA is essential in fields like cancer research, pharmacology, and gene therapy. As a future biochemist, I hope to contribute to the research that improves treatments and make medicine more precise and effective. This demonstration shows how even the simplest kitchen experiments reflect on the fundamental techniques used in modern biotechnology. By studying and understanding DNA, scientists are able to unlock solutions to the world's most challenging diseases. And that's what I hope to be a part of.